Okay, so now come on, quickly show me your beautiful faces and let's start today's class.
What's the bad news?
No, none. There's no bad news. Only good news. There's no bad. The bad news is, no, that's not a bad news, but the bad news is that we're going to start another botany chapter only. But there's left.
Plant growth.
Yeah. Yes, plant growth and development.
1 minute.
Okay. Are you back, Miriam?
Yeah. Sorry, someone came.
Okay. Alrighty. So let's start. So we were doing electron transport system, right? Now in that, now see, remember that I said that the complex 2 is also known as succinate dehydrogenase?
Yeah.
The complex 2 can also be called as FADH2. Okay.
Okay.
Now let us talk about the steps of the ETS. The first step was that the electrons from the NADH, it is produced in the mitochondrial matrix and during the citric acid cycle that we studied previously. Now those are oxidized by a complex that is known as NADH dehydrogenase. So electrons from the NADH is sent to the NADH dehydrogenase. Oh, just give me 1 second. I downloaded a video for you people. Let me just load it, okay? Just give me 1 second. I'm loading that video so that after I'm done, teaching you all the thing about ETS, then you can see it in the animation. Okay? Just give me a little while. I'm just. You do know that I can see you eating the cookie, right? Okay, now coming back to the topic. So the first thing that happens in the case of ETS is that electrons from the NADH comes to the NADH dehydrogenase. Okay, now after that,
What
electron.
about the video?
First I'll explain, then I'll show you the video because then only you'll understand it better. Because if I'll show you the video, just the video, you'll not understand. So first the theory, then the video. Now I haven't seen the whole video, so I don't know how much of an understanding that would be. I saw the best one on the YouTube, and
It's animation.
yeah, animation and dictation of whatever is happening. Maybe it'll be right, maybe not. Let's see.
Okay.
So the second step that happens, that electrons from here are then transferred from the NADH2 to the ubiquinone. Now this ubiquinone is located within the inner membrane of the mitochondria. These 2 points I think I've already told you people, right? Oh, now apart from this, what happened?
You wrote all the notes?
Only for this.
Oh.
I mean, I was like, I had time, and because I don't want to look weird, I was like, okay, let's just do it. Okay, because not the whole chapter. I didn't get that much of a time, but whatever I did, I did. Okay. Now, after that, what happens is that the ubiquinone also receives the reducing equivalence from the FADH2, which is the complex 2. Okay. And this is generated during the oxidation of succinate, meaning FADH2 was being formed in the TCA, remember, at one place only. So
Yes.
from there also the reducing equivalents were received and it went to ubiquinone. Okay, and ubiquinone is located within the inner membrane of mitochondria. Got it? Now, once the ubiquinone gets all the reducing equivalents, so it will get reduced. Okay,
Yes.
so it's like its form or name will be changed. So now the reduced ubiquinone is known as ubiquinol. Okay, now this ubiquinol, it gets oxidized with the transfer of electron to the cytochrome C. Now this ubiquinol, it transfers the electron to the cytochrome C via cytochrome BC1 complex, which is complex 3. So it is sending to the cytochrome C with the help of a carrier, that is cytochrome BC1 complex. Okay. Now later on, what will happen? After the electrons are coming onto the cytochrome C. So now cytochrome C is a small protein. Okay, it is a mobile protein, carrier protein. Okay, it is attached to the outer surface of the inner mitochondrial membrane. Outer surface of the inner mitochondrial membrane. Getting the location? Meaning like this is the outer membrane, right? This is the inner membrane, right? So on here,
Yes.
the cytochrome C is present. Outer surface of the inner mitochondrial membrane. And this cytochrome C, it would act as a mobile carrier. Mobile means anything that can move, has movability. Now, what it does is that cytochrome C, like this is a very commonly asked question, like what is the function of cytochrome C? So you would say that the cytochrome C transfers the electron in between the complex 3 and the complex 4. Okay, so just it's like doing this business, taking here, giving to them, taking here, giving to them. Okay, so it's sort of acting like a mobile carrier. Got it? Do you have a test today also?
I did the test.
Oh, how did it go?
The cell was nice.
Which chapter?
Cell.
All right, now talking about the last point of the whole ETS cycle is that oxygen acts as the terminal electron acceptor. The last one to accept the electron is oxygen.
You are correcting my papers.
Yeah.
So everything is correct.
Am I correcting your paper or am I setting your paper?
No, are you correcting my papers?
I just corrected one.
What about the animal kingdom, morphology?
The animal kingdom is corrected by me only.
Morphology?
Wait. Morphology, yeah, that I'll be correcting.
Everything is correct. Why didn't you cut marks?
Because everything is correct. I did cut marks. You did not get full marks.
For me, they corrected everything. They were like, good, 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 good. All things are, there's something,
Yeah,
concepts are clear with something, something.
yeah. So, like, why are you doubting yourself? If I have corrected it, means your concept is clear. Why do you have a problem with that?
I thought they were just randomly correcting papers.
No, no, no. See, I have written where you haven't written the enough facts. I have written more question mark. Haven't you seen that? Have
Oh,
you have you even looked at your paper?
and I
Corrected?
looked, but then I just saw good, 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 good.
No. And see, I did not actually see. The main thing is that you wrote vaguely. Okay, but whatever you wrote, even after being vague, was right. So I was like, okay, now how can I cut marks where that is
Which
being
animal kingdom?
hmm? Morphology, I'll check today. I haven't checked it yet.
Mm.
Now see the last point that says that the last acceptor of oxygen, the terminal one, the ending part is by oxygen. Okay, oxygen is the last electron acceptor. Now, as soon as it accepts the electron, it becomes reactive and then it combines with proton to form metabolic water. So at the end of ETS, oxygen, like this question also arises, like which molecule acts as the terminal electron acceptor? Oxygen. What is formed at the end of ETS? Metabolic water. Okay. So ETS is clear?
Can you show me the fifth point?
Fifth or sixth?
Fifth. In fifth.
You'll watch the video, you'll know what this whole thing is about. Okay. Say what?
Fifth and sixth, are they connected? They're not connected, right?
No, they're not connected. It's like electrons are being transported to another, and at the end, like once the electron has been transferred between complex 3 and complex 4, then the ETS is done because there are only 4 complexes, right? And then from complex 4, it would go to oxygen, which is actually acting like the last one to accept the electron.
Okay.
Now, all of you, like both of you, there are only 2 people in the class, you read it yet again. Okay, you read it. No writing it right now, only read. Because right now you'll see the video. Then you can note down. Okay. You read all the points, fix it in your brain, and then we'll see what happens in the video. I'm scared to play the video.
Why?
Because I'm like, okay, what if it confuses you even more? We'll see. If I think that will be doing, we'll stop it then and there. Okay, you read it once mindfully, then I'll play the video for you.
What is NADH2?
NADH2 was the energy intermediate. NADH2, FADH2, like how ADP, ATP is there, chemical intermediates. Yes.
So where was this NADH2?
TCA. Do you remember 3
Oh, from
points?
there it's coming.
Yeah. From TCA now, whatever electron, whatever NADH, these are all connected to each other. Okay.
What is the difference between NADH and NADH2?
NADH2 is formed when NADH gets reduced.
Oh yeah. So in the first point, electrons from NADH?
See, here you just read the points. You should know what is happening first, what is happening second. When you will see the video, you'll know what is actually happening, how the electrons are being transported. Just read the sentences. Tell me when you're done so that I can scroll. Read it mindfully, not like blah, blah, blah. Khadija, you're also reading it, right?
Yes, ma'am.
Third point.
It is good.
Next point. Done.
Both of you?
Yes, ma'am.
Let me just. Okay, just give me 1 second. Okay. Can you see the screen?
Yes.
Yes, ma'am.
The queen.
Can you hear the audio also?
No.
No.
The audio is not there?
No,
No.
ma'am. Mm.
Why is the audio not there? Okay, just wait. Give me 1 second. I don't know why the audio is not working. Now the audio is there?
No. No.
No? Okay, then watch it without the audio only.
No way. In Zoom, when you share the screen, there will be an option.
Where?
I don't know where, but before it used to be there.
Okay, there is. I'll have to see. Where would the option be? If
When you share the screen, right? You have like many tabs will come, right? And then below that, is there an option?
Got it. Share sound. Okay.
Yeah.
Electron transport
Audible now?
chain or
Yes,
respiratory
perfect.
Yeah.
chain.
Okay, now let's watch it. Switch on your videos.
It is present in the inner mitochondrial membrane. The electron transport chain consists of various electron transport or electron carrier molecules. The electron transport molecules are arranged in a sequence. They carry or transfer electrons from reduced coenzymes like NADH, FADH2 to the final electron acceptor, that's oxygen. The electron transport molecules are known as components of the respiratory chain. Some of them are proteins. However, non-protein carriers are also present. The position of a particular component in the respiratory chain depends on its redox potential. The components of the respiratory chain are arranged in the order of increasing redox potential. Starting compounds have negative redox potentials, and terminal components have positive redox potentials. Therefore, in the respiratory chain, electrons flow from negative to positive. In the respiratory chain, electrons flow from NAD to cytochromes via coenzyme Q, and then from cytochromes to molecular oxygen. Coenzyme Q also collects electrons from FAD. The transfer of electrons from substrates to NAD and FAD is catalyzed by dehydrogenases.
Do you see that this cytochrome C?
Yeah.
This cytochrome C is the mobile carrier. It is transferring in between the third complex and the fourth complex. Do you see it going this way, oscillating it to and fro?
Yeah.
Electrons from FAD.
Taking from here, transferring.
The transfer of electrons from substrates to NAD and FAD is catalyzed by dehydrogenases. At the electronegative end of the respiratory chain, NAD linked to more positive. Others are NAD, coenzyme Q, cytochrome C, and oxygen. All the complexes are integral membrane proteins. Each complex is involved in oxidation and accepts electrons from mobile electron carriers and passes electrons to another mobile electron carrier. Complex 1. Complex 1 is NADH coenzyme Q reductase or NADH dehydrogenase. FMN and iron sulfur clusters are prosthetic groups of this enzyme. Electrons collected by NAD are transferred to coenzyme Q by NADH coenzyme Q reductase via FMN and iron sulfur cluster. So NAD is oxidized. Complex 2. Complex 2 is succinate coenzyme Q reductase.
Succinate
It also contains,
or you can call it as FADH2 also.
FAD and iron sulfur clusters. Complex 2 transfers electrons from succinate and glycerol 3-phosphate to coenzyme Q via FAD and iron sulfur cluster. Complex 3. Complex 3 is coenzyme Q cytochrome C reductase or cytochrome reductase. It also contains iron sulfur cluster. Complex 3 transfers electrons from coenzyme Q to cytochrome C via cytochrome B, C, and iron sulfur cluster. As a result of this, coenzyme Q gets oxidized. Complex 4 is cytochrome oxidase. It transfers electrons from cytochrome C to a final electron acceptor, oxygen. Hence, cytochrome C is oxidized.
Did you get anything? Is it a little bit clearer or still at the same position?
It was
clearer before.
no.
I think it would have helped you to imagine how the electrons are actually being sent across the whole complexes, right?
Yeah.
Yeah, but
The names were different.
Yeah, see, the names were different according to, you can say, a higher level. But at our level, whatever the names are there, the complexes names, we'll use that only. The thing that is happening there is the same. Okay. So that is your ETS. Now tell me if you have any doubts, because there's nothing extra or anything apart from this that I can, explain to make you understand this, because this is the only thing that happens over here. Okay. And at the end, the oxygen takes the last electron and it forms the metabolic water. And as soon as the metabolic water is formed, then the whole process of ETS is like coming to an end. Okay, now after this, we'll be talking about after I explain this. Okay, now right now we will be talking about the oxidative phosphorylation. We saw substrate level phosphorylation, right? Now we'll be talking about the oxidative phosphorylation. Now, what is phosphorylation?
Formation of ATP.
ATP, right. Now we'll see how it is done with this mechanism. Now see, this one you might be familiar with, chemiosmotic hypothesis. Photosynthesis, chemiosmotic hypothesis. Remember? See, now what happens is that when the electrons pass from one carrier to another via the complex 1 to 4. See, electrons are transported to each and every carrier with the help of the complexes that are there. Okay, now this happens in the electron transport chain. Now, all of these electrons, they are coupled to ATP synthase. Now, ATP synthase is the complex number 5, the fifth complex. Now, ATP synthase. Okay, this is the name of the complex. Now, when electrons are sent to the ATP synthase, why they are sent there? So that the ATP can be produced from the ADP and the inorganic phosphate. This is what happens. Okay. Now, we already know that for the formation of ATP, like how we studied, a proton gradient is required. Isn't it? Do you remember in the lumen of the thylakoid, all of the protons were being collected, and when that gradient was broken, then the ATP was synthesized, right? This was the chemiosmotic hypothesis. Now, the proton gradient required for photophosphorylation is obtained with the use of energy of oxidation and reduction, and therefore it is known as oxidative phosphorylation. And this is the same thing which is explained by P. Mitchell through the chemiosmotic hypothesis. How the ATP is formed, how, look over the diagram here also. Here proton is accumulating and proton only is going through the ATP synthase. Isn't it? From F0 to F1, and then in that process only ATP is synthesized. Getting my point? What is the confusion? Tell me. Now see, ETS, electrons has been transported everywhere wherever it is needed to. Now ultimately, what is the sole purpose of doing any of these like activities that are happening?
ATP.
Yeah, ATP, energy formation, right? So after ETS, this phosphorylation takes place. And this is known as oxidative is because oxidation and reduction, both the processes are incorporated in, meaning used here. Now, inner mitochondrial membrane is permeable to protons. Okay, inner mitochondrial membrane, I cannot see my cursor. It is permeable to protons, but only in the region of F0 to F1, right? This part. Here only it is permeable to protons. Okay. Now, the energy released during the ETS is utilized in synthesizing the ATP with the help of ATP synthase, that is the complex 5. As I told you, the ultimate goal is to create ATP so that energy is created. Now, this complex, which is ATP synthase, we studied about 4 complexes, right? Now we're talking about the 5th complex, that is ATP synthase. It has 2 components. The first is F0 and then there is F1. F0, F1 are the 2 components of this particular complex. Okay, now talking about the F1 is the head piece and it is at the peripheral position. Okay, and the F0 is an integral membrane protein present inside. You remember integral and peripheral protein we studied in cell, plasma membrane? Yes. So whenever the protons would pass this complex, ATP will be synthesized. Meaning whenever protons would pass the ATP synthase, that is the complex number 5, then the ATP will be synthesized. Now see, the passage of protons through the channel is coupled to catalytic site of F1 component for the production of ATP. For each ATP produced, 2H plus, meaning 2 protons pass through the F0, meaning from the F0 it goes to the F1, 2 molecules of protons, and then ATP will be synthesized. What happened? Tell me.
The passage of protons through the channel is coupled to catalytic site of F1.
Don't go with the language. Just try to understand the concept that I'm saying. Okay, don't go with the language. Let me see what NCERT says. See, basically, let's go according to NCERT. Okay, strictly to NCERT. What happens is that there is a complex 5. The 5th complex is there, which is known as ATP synthase complex. Okay. Now, this complex contains 2 components, F0 and F1. Okay. Now, the F1 headpiece, it is the peripheral membrane protein, meaning it is present at the periphery. Look here, it is shown in the diagram only, isn't it? It's present at the periphery, whereas this one is present in the integral part, right?
Yes,
inside.
Yes.
Okay. Now, the F0 is integral membranal protein, right? Because it is present in the inner membrane. This much is clear?
Yes.
Now, when the protons pass through this channel, meaning F0, F1, or I can say it passes through ATP synthase, which is the complex 5.
Yes.
Now, during that process, ATP is synthesized. Okay. Now, in order to synthesize 1 molecule of ATP, we require 2 protons.
Okay.
Okay. Now, that line is written in NCERT also. The passage of protons through the channel is coupled to catalytic site of the F1 component for the production of ATP. Now, the meaning here is that for any reaction to occur, there has to be a site of that reaction to occur. Okay. And since ATP synthase is that complex that is happening, that is making the ATP, it has certain catalytic site through which when the protons will go, ATP will be formed. This is what the line says. You do not have to write it in your exam also. It doesn't matter. You should just understand what it says. Okay. And then ATP is produced after 2 protons are passed. This is very important to remember. ATP is produced when 2 protons are passed. Okay. And
1 molecule of ATP is produced.
One molecule of ATP. When 2 protons are passed, first it will pass through the F0. Then it will go to the F1. Okay. So when 2 protons passes through the F0, now where is F0 present? So it is present in the intermembrane space. This particular thing, this one, it is the intermembrane space. Meaning the space
integral.
Yeah. Integral is inside, right? So the space between the outer and the inner is the intermembrane, right?
Yeah.
Inter means between 2.
Yeah.
So the part of the ATP synthase present in between the 2 membrane is F0. The one that is present on the periphery, meaning on the peripheral side, is the F1. Any doubts?
No.
So what was the difficulty over here? It is the same thing written.
No, you explained it better.
Yeah.
Is it clear?
Yes.
Yes.
So now you people understand how many protons are required to make how many ATPs?
Yes. Can you explain again the second point?
Proton gradient.
Yeah.
See, whenever the electrons are circulated either through the photosystems, photosystem 1, photosystem 2, right? Or through the ETS. Okay, there are electrons also made during the processes, and then there are protons also synthesized. So electrons, they go to the ETS for them being converted into metabolic water later on. The electrons will get converted into water later on, and the protons, which were formed as a result of all these reactions, photosystem 1, photosystem meaning cyclic, non-cyclic, and all of these. So these protons, they start accumulating towards the lumen of the chloroplast. Lumen is just like the area enclosed. Okay, we're talking about plants. We're not talking about animals. Now, after the protons have accumulated in the lumen, then what will happen is that this complex, which is a complex 5 ATP synthase, now when the protons would pass through this complex, then ATP would be synthesized. And this whole theory that I just said, this is chemiosmotic hypothesis, meaning this is a hypothesized way that, okay, this is how it will happen. And it is given by Peter Mitchell. Any doubts?
Then what is oxidation reduction?
Oxidation reduction is like they're saying why the name oxidative phosphorylation has been given over here to this mechanism. Okay, see, the main reason is because, you know, it is happening after the ETS. Okay. And since oxidation and reduction, both the process are involved in this, that is why it is oxidative phosphorylation. Is it clear now?
Yes.
Very good. Now, do you remember when we were discussing in the photosynthesis chapter, I said that one NADH forms 3 ATP, this line. One NADH forms 3 ATP, one FADH forms 2 ATP.
Yes. So
we were like, okay, when 3 NADH is there and we run 6 cycles and we have 18 ATP. Remember that? Yeah. So this is what this line is saying. Okay, it is the same thing because it is all connected. Got it? Any doubts up till now?
No.
Sure? You have a doubt? Okay. All right. Now you people can note down. From
only
point number 3.
4. Done.
Harira, you're also done?
Yes, ma'am.
Oh. I will still suggest you people to note this part also with the drawing. Because once
The
you
book.
learn
The book thing.
yeah. Because all of these points, when you'll ponder it, you'll find it very easy to understand, okay? So you note those down also.
Can you zoom in a little bit? What did you say the name of this process?
Name of
Oxidized.
which? Oxidative phosphorylation. Oxidative. Oxidized phosphorylation. What happened? Spaces?
Can I just write proton gradient is required?
Definitely. Just don't write this particular point. The 7th one you don't need to write. Rest all you copy down. Okay. These things
Okay.
that I have written in my book, this also you don't have to copy down.
Can I also skip the point? Mechanism of membrane linked ATP synthesis is explained by chemiosmotic.
No. You can't skip that. That is in the syllabus. It's important. It's there in the previous chapter also.
Okay. In the 6.6, can I just write from for each ATP produced? Can I just not write the coupled thing?
Okay. Yeah. Skip that. You write from for each ATP produced.
Done.
Both of you are done?
Yeah.
All right. Now, do you people know that, like, in general, I'm asking, like, for those people who have had, like, commerce as their stream, do you know that they get to make their balance sheet something in accounts? Do you people have any idea about that? No? Okay. Anyway, even if you don't have any idea about that, we will make a balance sheet today. Okay. But we will make a balance sheet for ATP. We'll see where ATP is made, how many molecules are made, and in totality, how many are made. Okay. Now, NCERT says that for, okay, and we'll call it as, wait.
Shall I ask my sister?
It's all right. You can ask your sister, what is a balance sheet?
Yeah.
She has commerce as a stream.
She took 10 years ago.
It's okay. Don't bother her. Let's see what the thickest one does. The respiratory balance sheet.
When will this chapter end?
I was thinking the same thing. Hopefully today. All right. Now, talking about the respiratory balance sheet, see, whatever is happening, because we are studying respiration in plants, right? Meaning this whole chapter revolves around the term respiration. Now, in all of these things, we have to see where ATP is being synthesized, how many ATP molecules are being synthesized, and in how many cycles and everything that we will see under this. Listen, now, the main thing to remember over here is that the situation or the steps that we talked about, like we saw glycolysis, we saw ETS, we saw phosphorylation. Okay. All of these things would happen on their own. Okay. But for our ease of understanding, and so that we can make this balance sheet, we have to actually assume that they are happening one after the other, sort of like in a series. Okay. Just like how we can say glycolysis is followed by TCA. TCA is followed by ETS, right? So we'd say glycolysis will be followed by the TCA. This, this wherein one would stop, the other would start. This does not happen in an ideal condition. Like in normal way, this does not happen. Like one is not followed by the other. But for the
TCA is Krebs cycle.
Tricarboxylic acid cycle. So for the ease of our understanding, we are just assuming it. How like in equations we say let X be this. So for now we are saying let all of these steps follow each one. Okay, so meaning one is happening after the other. Got it? Now the second point that we have to consider while making the respiratory balance sheet is that the NADH, which is synthesized, it is actually particularly, you can say, synthesized in the glycolysis. Then that NADH is transferred into the mitochondria. And then in the mitochondria, oxidative phosphorylation takes place. These are all the things that I'm reading, that you take out your NCERT book, page number 234. See all the points. Look at all the points given in the book.
Yes.
Yeah. Now see, these are the stuff that we are actually assuming in order to get the whole balance sheet. Okay, we are making an assumption over here. The first assumption that we are making will be that all of these steps will be taking place in a serial manner. Okay.
Okay.
Getting my point? So all of these steps will be taking in a serial manner. Secondly, the next assumption that we are going to take is that the NADH, which is synthesized in the glycolysis pathway, it is transferred to the mitochondria, and after that it undergoes the oxidative phosphorylation, meaning the formation of ATP.
Okay.
Getting my point?
Yes, yes.
Then the next thing that we are assuming here is that none of the intermediates, okay, meaning the NADH, the FADH, all of those, they are not utilized. But this is not possible, right? They have to be utilized.
Yeah.
So yeah, so this is an ideal situation we are taking in. And the last assumption that we are doing is that only glucose is being respired and no other substrates are entering the pathway, which is again not possible. Only one molecule of glucose being respired is not possible, right?
Yeah.
So these are all the assumptions that we will be making in order to make the ATP balance sheet. Okay? Because without this, we cannot make the balance sheet. Got my point?
Yeah.
All right. Now, let us move forward and look at what are the things that we will be making. So, the heading goes ATP molecules produced during respiration. ATP molecules produced during respiration. Now, first you make a column known as stage of respiration. Stage of respiration. The next column goes source. And the third column, number of ATP molecules, number of ATP molecules produced. Okay. These are the three columns you have to make. Now, under the column of the stage of respiration, you write first one is glycolysis, because from here only the whole thing started. So first we are talking about glycolysis. Now, directly during the process of glycolysis, directly 2 molecules of ATP are made, right? And during the process of glycolysis, 2 NADH2 are made, right? Now, from these 2 NADH2, how many ATP will be made, if you remember?
3. 6.
How many? 8? Yeah, 6. Because from 1 NADH2, how many molecules of ATP are made? 3 molecules. 3. Very good. So you can say 1 NADH2 makes 3 ATP. So that you know that 2 into 3 is equal to 6 ATP they are made. Now, another step or another stage is the second one is pyruvic acid to acetyl coenzyme A. Do you remember link reaction?
No.
You don't remember? Link reaction wherein the pyruvic acid formed after the glycolysis was pyruvic acid was made into acetyl coenzyme A so that it can be entering the citric acid cycle. Now you remember?
Yeah.
So during this process, 2 molecules of NADH, how many ATPs will they make?
6.
So they will make 6 molecules of ATP yet again. Now, after that, the third step, the last one, is the citric acid cycle. Okay, now here, you can also write TCA cycle or you can write citric acid cycle. Now, the source, first of all, 6 NADH2 are made. Then how many ATP will be made?
18.
18, because 6 3s are 18, right? Then 2 FADH2 will be made, like in the TCA cycle. So how many ATP will be made? FADH2. 1 FADH2 makes how many ATP?
4.
1 FADH2
One
makes
FADH
4.
makes 2 ATP, so 2 FADH will make 4.
Very good. So 1 FADH2 makes 2 ATP. So 2 of them would make 4 ATP. Very good. Now at the end, directly also some ATP molecules are synthesized and the number is 2. So ultimately, can you count how many of them? 2, 4, 18, 6, 6, 2. How many?
30.
Eh? 30?
30. What did you say? What did you ask
18.
me?
I'm asking you the total number of ATPs now.
Yeah, total number of ATPs. 18 plus 2, 20, and then 20 plus 6, 26, 26 plus 4, 30.
How is it coming 30?
Oh, wait.
How is it coming? That is what I'm saying. How can it come 30? It can never come 30. It can either come 36.
Wait, show me the second one. For the second one, why did I write 18? Oh, I wrote it. What did I do?
Didn't you write it like this? See,
Wait.
I just wrote NADH to this part over here only, like how many it makes. Now here I didn't. So here it is pre-assumed.
Link reaction, that. Oh, okay. I have to cut over here. I have to write 6. And the third one, TCA cycle.
Now how many molecules?
Can you scroll a bit? Can you scroll a bit? What is direct?
Direct is like not through NADH, but direct ATP synthesis, meaning when ADP turns into ATP, this thing. This is direct. Meaning
Okay,
no
so right
NADH.
now, 36.
38? How 36?
Oh, you're adding the direct one too. Okay.
All the ATPs. ATPs are ATPs. So all the, like you can say, in totality, net gain of ATP will be 38, right?
Yes.
Yes. But another thing you have to look at over here is that during the process of this thing, like in the most of the eukaryotes, okay, this is one line that you must remember, that in most of the eukaryotes, the net gain of ATP will be 36. Okay. It's equal to 36 molecules. Got it?
How 36?
Is because anaerobic respiration does not take place in the case of eukaryotes. That's why.
What is anaerobic?
How does glycolysis happen?
Oh, so the direct one won't happen.
So ultimately we'll have the 36 molecule. Okay, but yet again this is an assumption. Okay. And like if we are talking about the aerobic respiration, mainly during the aerobic respiration, 36 ATP molecules are synthesized in the case of eukaryotic cells. Got it? So this is your respiratory balance sheet. Okay.
In eukaryotes, aerobic respiration.
Only 36
So,
molecules of
38 ATPs molecules will be.
See, 38 ATPs are for the whole respiration process. We are not considering like whether it is aerobic, whether it is anaerobic. But if we are considering only aerobic, then it will be 36 molecules.
Okay.
Okay.
Done.
Any doubts?
No.
Sure.
Yes.
All right. Now coming on to the last topic, or the second last topic of this whole entire the whole entire chapter is the amphibolic pathway.
Again, pathway? How many pathways? How many pathways?
Amphibolic pathway. Now, before you try to understand what amphibolic pathway is, I'm sorry, my handwriting is really bad because my hand is not working because my hand is frozen.
Why is it frozen?
Because it's so cold over here.
Oh, my hand is burnt.
Your hand is burnt.
I burnt my hand yesterday.
Oh, you really did?
Yeah.
How?
I was about to straighten my hair.
Oh God.
And then I just touched the part where it was hot. Like I grabbed it by that. So like I have like a blister kind of thing over here.
Okay, now first of all, you people answer this question. What is the most favored substrate used for the respiration process? Substrate. It is the most favored one. You people have completed the whole entire chapter, almost coming to the end of it, and you people don't know what is the favored substrate for respiration. Have you heard of the very unique name known as glucose?
Yes.
Have you heard of glucose? See, substrate is anything on which the enzymes or anything, the reaction that takes place, it happens because of the substrate only. So how does the whole process of respiration starts? It starts with glycolysis. What is broken down in the process of glycolysis? Glucose. So the most favored substrate for the process of respiration is glucose. Always remember this particular point. Okay.
I don't understand.
See, meaning there are so many substrates. We saw that sucrose was there. Why didn't sucrose get into the glycolytic cycle? Why?
Because nothing happens to it.
Yeah, because it is inert. So the
Yeah.
sucrose had to be converted into glucose. And glucose is that one beautiful molecule that will happen, like enter so many reactions and so many products will be made. This is the reason why it is favored. It is favored because it reacts well. Got it?
Okay.
Any doubts?
No.
So for this very reason, since glucose is the favored substrate, so what about the other carbohydrates? So all those other carbohydrates which are there, they will be converted into the glucose only. Okay? They will be converted into the glucose molecule, and then only it would enter, or you can then only it will be used for respiration.
Can you repeat?
There are many carbohydrates in the world. Fructose, sucrose, galactose, so many of them are there.
Yeah.
So all of those carbohydrates, see, carbohydrates are used to derive energy, right? Energy giving.
Yeah.
So in order for them to give energy, they have to enter, they have to be respired. Respiration has to happen, then only we get the energy.
Yes.
So all the carbohydrates, first they have to be converted into the glucose, and then only they can undergo respiration. Because glucose is the most favored substrate for respiration.
Okay.
So all of them would be, actually, first of all the carbohydrates, they have to be converted into glucose for the process of respiration. Is this line clear?
Yes.
So we'll write all carbohydrates are usually converted into glucose before they are used for respiration. Before they are used for respiration. Okay. Now, apart from glucose, apart from carbohydrates, there are so many other molecules also, like fats are there, right? Proteins are there. So, see, all of these substrates can also be performing in the respiration, but they don't directly enter the respiration process. See, other substrates. Let us write the example of other substrates. So, wait. Other substrates. What they can be? So we can have fat as the substrate. We can have proteins, right? So these are also substrates only, and they can actually enter the respiration process, but not directly. Okay, they have to be converted into certain stuff to enter the respiratory pathway. Getting my point? So now, talking about the fat, the first one we talk about is fats. Now, what happens to the fats is that they are broken down into glycerol and fatty acids. Okay, so talking about the fats, so they are broken down to glycerol and fatty acids, because see, fats are made up of glycerol and fatty acids. So when you break down the fat molecule, you will get glycerol and fatty acid. Okay. Now, these fatty acids, the fatty acids are converted into acetyl coenzyme A. Okay, so the fatty acids, you can say, degrades into acetyl coenzyme A. Now, can you tell me, acetyl coenzyme A, can it undergo the process of respiration?
Yes.
What kind? It can undergo which kind of
Aerobic.
respiration? Yes, aerobic respiration. So ultimately it is undergoing respiration only, right?
Yes.
So fatty acids, they get degraded into acetyl coenzyme A and then enter the respiration pathway. This much is clear?
Yes.
So when the fats are converted into the acetyl coenzyme A, then only they can enter the respiratory pathway. Got it?
Yes.
Now, talking about the glycerol. Now, this glycerol, this is like, first we saw how fatty acid enters. Now, let us talk about the glycerol. So, the glycerol, they would be converted into PGAL, phosphoglycerol aldehyde. So, glycerol is converted into PGAL. You're pagal. Yes. You're very famous pagal. So, phospho, you write down the full thing. Don't write pagal. So, phosphoglycerol aldehyde. And when it will get converted into phosphoglycerol aldehyde, then it can enter the pathway. These three things are clear?
Yes.
In order for the fats to enter the respiratory pathway, first of all, it has to be broken down into glycerol and fatty acid. The fatty acid will degrade into acetyl coenzyme A, meaning it will get converted into acetyl coenzyme A, and then it will enter the respiratory pathway. What about the glycerol? So glycerol will be converted into phosphoglycerol aldehyde and then it will enter the respiratory path. This much is clear?
Yes.
All right. Now let us talk about the second type of substrate, that is the protein. Proteins are made up of what?
Amino acids.
Very good. So proteins, they are converted into amino acids. Now, amino acids, since we are converting them into amino acid, means we are breaking the protein, right?
Yes.
We are degrading the protein. So, they are being degraded. Okay, so they are being degraded. And then what will happen? They will enter the respiratory pathway, meaning the amino acid will enter the respiratory pathway. Okay. Is it clear up till now?
Yes.
All right. Now moving forward, this was about proteins. Any doubts?
No.
All right. Now see, amphibolic is like a term which is used for like both the terms. See, do you know that metabolism contains, if we talk about metabolism, you people understand? So metabolism contains anabolism plus catabolism. Do you know this thing or not? Yes or no?
No.
All right, I'll tell you what it is. See, anabolism is the process in which molecules are synthesized, okay? And catabolism is the process wherein molecules are broken down, okay? Anabolism is the process where molecules, any molecule, if we are talking about any molecules being synthesized, so that process is anabolic process. Getting my point?
Yes.
So if I say glucose is being synthesized in photosynthesis. So we can say that photosynthesis is anabolic.
Yes.
Now catabolism means molecules are broken down. We can say that respiration is a catabolic process.
Yes.
But yet again, there is another unique thing to it, that if we talk about respiration, okay, or the respiratory pathway, so the respiratory pathway can be considered as amphibolic. Okay, so talking about the respiratory pathway.
So it has both anabolism and catabolic.
Yes, exactly. So talking about the respiratory pathway, it is amphibolic. Amphi means dual, right? Like
Yes.
amphibians, you understand?
Yes.
So it is amphibolic pathway. Amphi means dual. And it contains anabolic plus catabolic. Okay?
Yes.
Clear to everyone?
Yes.
Great. Now, so this was about respiration being amphibolic pathway. Now coming on to the last topic of the chapter, that is respiratory quotient. Respiratory quotient. Now, talking about the respiratory quotient, see, what happens is that during the process of aerobic respiration, okay, oxygen is consumed, meaning we use oxygen and CO2 is released, isn't it? Because aerobic happening in the presence of oxygen. So we are using the oxygen and ultimately we are making the CO2. So when we want to find out what is respiratory quotient means RQ. Okay. Now RQ is the volume of CO2 evolved, meaning how much of the CO2 has been evolved after the process is done, and upon the volume of O2 consumed. So it means that how much oxygen is being consumed during the aerobic process that would lead to the evolution of the carbon dioxide. Okay. Now this respiration quotient will vary, like the value will vary depending upon whatever substrate that we are using. Okay. So we can say that the RQ value varies depending upon the respiratory substrate used. Okay, meaning what are the following respiratory substrates that we talked about? We talked about carbohydrates, we talked about fat and protein, isn't it? So the first case scenario, when carbohydrates are being used. Now when carbohydrates are being used, in that case, if we talk about, like, let us look at the equation. Okay, so C6H12O6, this is glucose, which is a carbohydrate, plus 6 molecules of O2. We are using 6 oxygen molecules and we are giving out 6 CO2 plus 6 H2O plus energy. I mean, we are not giving out, the plants are. So talking about the RQ of the carbohydrates will be 6 CO2, right? How many CO2 are evolved upon how many O2 are being used? What will be the answer? 1, right? Because this will be cut, isn't it? What happened?
Carbon dioxide and okay.
The formula for RQ goes volume of, or the amount of CO2 which is evolved. meaning given out. Like how we give out carbon dioxide when we breathe and we take in oxygen. So depending upon that, we can find out the respiratory quotient. Okay.
Even O2 will be cut.
Yeah, C will be left. Okay, now talking about when fats are used. Okay, when the substrates are fats. Now, they have given the example of tripalmitin, I think so. Let me check in the NCERT. Have they given? Yeah.
How
See.
is one left if carbon dioxide by O2 is left? So
We are calculating the number, not CO2 and O2. The number. How can you calculate C and O? The calculation is done for numbers. That is why we are getting 1. Okay. We are not doing this. O2, O2. No, we're not doing it. Okay. All right. Now, when we are talking about the fats, so tripalmitin. See, this tripalmitin is one kind of fatty acid. Okay. So we will be using tripalmitin. Now, this you don't have to remember. The reaction is not necessary to remember. Imagine how many H is there. See, 98 H molecules. Plus 145 O2 gives us 102 CO2 plus 98 H2O plus energy. So this reaction you don't have to remember, it won't be asked, but this molecule is tripalmitin, and tripalmitin is a fatty acid. Okay, it is a fatty acid. So now if we try to find out the RQ, what will we write first? How many CO2 is made? 102.
102
right?
CO2 by 145 O2.
So we can find out here that it will come out to be 0.7. If you divide it, you will get 0.7. Okay, so we can say that RQ is less than 1, isn't it?
Yeah.
Right, because RQ comes out to be 0.7. So the main thing to remember over here is that when fats are used as a substrate, the respiratory quotient will be less than 1. When carbohydrates are used, then the respiratory quotient will be 1. Okay, now the last one. Talking about the third case scenario, that is when we are using proteins. Okay, now in the case of proteins, the RQ value will be 0.9.
Where is the thing? Reaction?
It's not needed, it's not given.
Okay.
This is only,
so if this is less than 1. Even this is less than 1.
Yeah. So for protein it will be 0.9. Got it?
Yes.
Okay, now there are other two things also. They are usually asked in the like MCQs sometimes. So when we are using, you remember oxalic acid, oxalic?
Yes.
So when oxalic acid is used as the substrate, then the RQ which is there is 4. Okay.
And it is more than it.
Yeah, it is more than 1. And when we are using this thing, malic acid, you remember?
Yes.
So when we are using malic acid as the substrate, then the RQ is 1.33. Okay?
Okay.
Now, another thing here to remember, like, okay, we try to find out the RQ for aerobic respiration, isn't it? Now, what if we try to find out the RQ for anaerobic respiration? What do you
There
think?
will be no O2.
Yeah. So if we try to find out the RQ, so whatever, let's just say any value of CO2 is there, divided by 0 of O2, and anything that is divided by 0 becomes infinity, right?
Yeah.
The RQ value for anaerobic process will be infinite.
Okay.
Any doubts up till now?
No.
So with this, we have completed the chapter that is respiration in plants. Now tell me, how do you people feel?
There's
Happy?
so many things to learn.
Yeah, there are so many things to learn, and yeah, your test will be done for this chapter also, right? So while you are trying to study, just focus on the points that are there in the NCERT. Okay? And like
Okay.
nobody will ask you to write down the whole steps of TCA. Okay, that those kind of questions don't come.
Really?
Yeah, I don't think that kind of questions will come. Rather, what?
So they won't ask, describe the process cycle, Krebs
No,
cycle or
they will
something?
ask you what is Krebs cycle. They will ask you why it happens, where it happens, how it happens. They'll ask you what is the first stable product. But they'll not ask you to write down the whole cycle because you haven't been taught the whole cycle. Isn't it?
Oh,
NCERT
then
just
what about Calvin cycle and all?
Calvin cycle will definitely be asked. Calvin cycle is there in photosynthesis, 100%
Then
it will.
glycolysis also.
Yeah, glycolysis can be asked. But
Okay,
the name of the enzymes that I have told you, the main important enzymes, those can be asked. Apart from that, no enzymes can be asked. Okay.
okay.
So basically, we are done with this chapter. Now we're going to start the new chapter, that is plant growth and development. Okay. Now see, when we talk about the plant growth and development, here we'll have to see what type of things are involved in the process of like plants growth. Okay, and we'll also talk about what type of growths are there, what is the meaning of growth, and we'll see how the growth happens when we talk about the plant. So what are involved? So like hormones are involved. Okay, plant hormones. So we'll see how those plant hormones were discovered, what do they do, what is their function. All of these things we'll be seeing in this particular chapter. Now mind you, this chapter is much better, like happening nice, interesting chapter as compared to the respiration in plants. Okay, so today we're going to finally start the new chapter. Let me write plant growth and develop. Okay. Okay. Now today we are going to just discuss the basics of it. Okay. Now see, talking about the plant growth and development. First of all, tell me what is growth?
Growth is growth.
Growth is growth. Very good. Right. But growth is an okay. Can you tell me growth, is it is it a reversible change or an irreversible change?
No, irreversible.
Yes. Very good. Growth is irreversible and it is permanent. So any irreversible change or any irreversible permanent change in the size, in the length, in the volume, in the weight of an organism is known as growth. Okay. What are you saying?
Yeah.
Physically.
Physical? What physical?
Nothing.
Any irreversible,
I like this color.
me too, permanent change in the size, volume, or weight of an organism, weight of an organ or a cell, weight of an organ, weight of a cell, you just write weight of a cell, is termed as growth. This much is clear?
Yes.
Now, see, talking about, like, if you want to compare the growth of plant and the growth of animal, how would you do so? Like, what would, oh wait, I had to make you write another thing. I just forgot the difference. Oh wait, we have to go back to the chapter.
No.
One thing is left. One thing is left. Only one. We have to write down the difference between fermentation.
My book got over. I don't have pages to write.
Aerobic respiration. These are the two things that we have to find a difference in between. Okay.
Fermentation.
So we have to look at the difference in between them. So talking about fermentation, here what happens is that partial, I forgot this was there, partial degradation. Okay, partial degradation of glucose occurs. Okay, degradation means breakdown, right? You can also
Why
mention that
can't you just write breakdown?
you can write anything you want to. You can write breakdown also, or you can want to write, if you want to write degradation, you can write degradation. Talking about the aerobic respiration here, complete. I'm just writing complete, but you people will have to write. No, I'm also writing complete breakdown of glucose takes place. Okay. Here complete breakdown, there partial breakdown. Another thing that in the case of fermentation, net gain of ATP is equal to 2 molecules. Okay. For each glucose, we have 2 molecules of ATP synthesized. Talking about the aerobic respiration, 36 to 38 ATP are released, because we just saw the respiratory pathway, right? The balance sheet. Now, apart from that, in the case of fermentation, O2 is not required. And since O2 is not required, it is anaerobic. Talking about the aerobic, this is O2 is obviously required. So this is aerobic. Now, the next point says that the end product in the case of fermentation, what are the end products?
Lect.
Do you remember?
No. Ethanol plus CO2.
Yeah, alcohol is there and lactic acid is there, right? Because both
Yeah.
are the products of fermentation only. So the end products are lactic acid or alcohol. Now, if we talk about aerobic respiration, here the end products are CO2 and H2O. Okay. These are the following points you must remember about, like what is the difference in between the fermentation and aerobic respiration. Is it clear?
Yes.
Any doubts up till now?
No.
All right. Now coming back to the new topic, new chapter is plant growth and development. Okay. So for this, right now we are discussing growth. Now growth is obviously an irreversible permanent change. Yeah, I was asking you, What is the difference between the growth of a plant and growth of an animal? Like one thing, like let's just say if you want to compare the growth of us humans to the growth of plants, what is that one criteria you would take into consideration? What is the difference?
We
We
are living beings.
and plants are dead.
We can move, they cannot move.
I'm asking you the difference between the growth, not mobility, not what we can do, they cannot. No. Growth. One big difference, it's a very obvious difference.
Reproduction.
Growth, not reproduction, not processes, not body processes. I'm asking you the growth difference.
Let me think.
Khadija, what about you? Tell me. You people don't know?
Hint.
If you put a plant or tree beside you and you stand beside it, which one of either of you will be taller?
Tree.
Huh? Okay. Is there any other point in your life, maybe you can grow taller than the tree?
No.
Can you grow taller than a tree?
No.
Huh? Sure?
Yeah.
Yeah.
Sure, right?
Yeah.
So that's my point. See, the growth in the case of plants is unlimited. Whereas in our case, it is limited. We stop growing after a while, isn't it? Or not?
There was no way I could answer that.
So talking about the growth in plants, so the plant growth is unlimited. It like the plants keep on growing throughout their life. Whereas the case of animals, the growth in animals is limited. Meaning we will grow up till a point. The lion will grow up till a point. The lion will not grow as tall as a tree. Right? Have you seen a lion as tall as a tree?
No.
Even the trees are taller than the giraffe. Right?
Yeah.
Even the giraffe cannot surpass the tree even after being so tall. So this is one very basic difference. And I don't know how you people were not able to point this out. That the trees, they keep on growing throughout their life and we stop after
But they
a while.
stop.
No, they don't.
They do. You never see a tree which is like skyscraper tall.
Yeah, there are many trees. There is a tree, sequoia tree. It is so many feet high. It is actually 300 or 200 feet high. So there are many trees. See, the reason is that even if they don't grow as tall as a skyscraper, let's just say, but they'll still keep on growing. Our growth stops.
Yeah.
Right? This is my main point. Okay, not how tall the plant is or how tall the animal is. The main point is that the plants, they do not stop growing. As animals, we stop growing. Okay?
Yeah.
Yes. So let's just keep it till here only for today's class. Now in the next class, we'll talk about more things, differentiation, how division happens and everything. Okay?
Okay.